So, once again, a good day, my dear postgraduate students. This has been the slide for homework in our previous class. A slide for diagnosis justified. Sometimes it is too very simple that no further history is needed, and it is obviously on the screen. But what I am very much interested is, if you people could identify the tissue behind the lesion. Usually we are supposed to find out the lesion within the tissue. But here I am more interested in the tissue behind the lesion. And this is the sixth slide for Glide the Slide session. Welcome to it. What we had seen, and sometimes there might be a need for it. In fact, your eyes should be able to penetrate the slides. Many diagnoses can be made under the HNE one. A special stain can be an added advantage. And this is a PAS Alcian blue, making things more obvious thanks to the contrast. So I am finding long filamentous structures. I hope we have made the diagnosis. Whenever such structures are being seen, what will be important will be a clinical correlation. Look at this. It is not a normal endoscope at all. It is being virtually carpeted with a lesion. And in such cases, we know it is infective. The points to ponder shall be immunosuppression, chemotherapy, steroids. Whether you are able to see the yeast forms or the pseudo-hyphae or true hyphae. What is the differential diagnosis? And what other mycosis shall we be having in mind? And incidentally, I have pertinently put up another figure over here, wherein I am finding a figure of 8 or a budding. But I hope you people are able to make out the difference between this and our lesion. We have not yet come to our lesion. I am taking you back to the picture. So there is a PAS and a methenamine silver, which are able to make out the diagnosis crystal clear. I am finding long, filamentous, almost never branching fibers, slender. And there is a necrotic debris that is there. Other inflammatory infiltrate are not very obvious. Maybe under the HNE, I can make out a few inflammatory cells here and there. So the diagnosis is Candida albicans. In fact, albicans will be a higher diagnosis. We are overreaching. My professor, Dr. Ramesh Rao, used to call it as Candida species. So here again, this is the budding yeast form. I hope you are able to compare this with the previous one, wherein I am finding unequal or unequal budding. So one is large, the other is small. Whereas in the case of blastomycosis, both will be equal. So that is one. A second point in difficulty can be, this can be mistaken. Supposing I am seeing this cell alone, it can be mistaken for an RBC. Both of them will be colored eosinophil. And what are the structures I am seeing over here? I am finding there are multiple so-called segments over here. These are called the pseudohyphae. And there can be the blastospores and the chlamydospores. Now, these structures usually are seen only in two conditions. One, when there is a severe immunosuppression. Two, in a culture medium. Third one can be when it happens to be a contaminant in a tissue, so that it becomes almost like a culture medium. Now, what is the pseudo-hyphae? You find that the filament grows. Normally, we will be seeing this in the vaginal smear and all. But then there is something called as a pseudo-hyphae. When there is going to be an oral thrush or an aggression in the lesion, you find that it becomes a hyphus form and it causes a segmentation. Normally, we will not be able to see such septae. It grows and then it breaks. It breaks. But they are opposed to each other. They appear as pseudo. But if things go on all right, if it happens to be a culture medium or immunosuppression, then there can be a true hyphus that can grow, which can again accumulate the blastospores. So for any such organism, there are two modalities of division and multiplication. One can be the spore form, another one can be the pseudohyphus. And these spores are getting lodged elsewhere, they will be forming the yeast forms. And if congenial conditions prevail, it will grow into a pseudohyphus and the story gets repeated. So the diagnosis has to be Candida species. Candida albicans is a diagnosis of microbiology, which is better demonstrated by means of this. And this is a person. Surprisingly, he is not a microbiologist himself, and he is a French dermatologist called as Sabaro. And it is his dextrose agar that shows these something like cotton wool spots that I am seeing. We shall glide to the next slide. Now, my dear students, this is a lesion from a 65-year-old male with gradually increasing difficulty in hearing. The history you people will have to read carefully. Normally, as I told you, it is too much of a spoon feed when once the field is given as well as the history is also given. But let us get tuned to things better. Increasing difficulty, or it can be worded as hard of hearing. So this is the lesion that we see. Go back, do your homework, and we shall meet again in the next class. Thank you.